Good morning, everyone. It's great to be back. Good to see many familiar faces. And I'd first like to acknowledge Richard and the organisers for this brilliant conference, and also to acknowledge my colleague and co-director at NCNED, Sonia Marshall-Gradisnik, who's hard at work in the lab, which is running 24 hours at the moment, and the chair of our steering committee, Ross Humphries, who's here today. What I'm going to try and do today is tell you the story of where we've got to and why we've got there. I have the data we presented at the colloquium, and I'll touch on that, but it's important that you actually understand why we're going the directions we are and whether you actually understand the science of what we're doing. So that's really what I'm trying to achieve today. So if you were here last year, you would recall we were talking about a group of ion channels, calcium ion channels, called Transient Receptor potential. Clumsy name, but they do have a meaning, or TRIPS. And there's about 28 members of that in 7 families. And we've been focusing on the TRPM3 simply because of operational expediency. We can do that. But I don't want to limit the discussion just to the TRPM3, because when you see the data of the SNPs and what's happening in the DNA, in these ion channels, we're also seeing data of SNPs in other channels as well, and they do interact. You know, they form heteromers, they influence each other, so it's not a straightforward story, but we'll use this as an example. The second point I want to make is that we've been doing work in natural killer cells, which are diabolical to work with in CFS patients or any time, but I want you to understand this is not just about natural killer cell pathology, but rather we use that as an exemplar for what might be happening in every cell and every tissue in the body. The reason we do that is that if you stand up in a calcium conference or whatever and you say, well, we've shown these impairments of calcium signalling, the critics in the audience would say, well, that's very well and good, but what impact does that have on function? So the reason we use natural killer cells, because as their name suggests, they lyse or kill cancer cells, invading cells and so on, and you can measure that. So there's an objective measure of the performance of those cells under different conditions of calcium signalling. Now, just by way of context, the TRPM3 receptor had never been identified on immune cells before. We were the first in the world to do that, identified that on natural killer cells. Not only did we identify it in healthy controls, but also in patients, and as you will see, the expression of those receptors is greatly reduced in CFS-ME patients. And not only is the expression reduced, but the function is reduced. And when we talk about calcium signalling, you will see that there is an impact of that impairment of these receptors in terms of calcium signalling, and there's all sorts of very complex gymnastics we go through with that, and I'll touch on that as well. Now remember that calcium is one of the most important intracellular signalling mechanisms. All cells rely on some sort of signalling mechanism in the cell to make sense of what's happening in the external environment, you know, impacted by hormones and a whole bunch of other things. But those signals have to be converted to messages within the cell that then manage a whole lot of intracellular functions. These are biochemical functions, they're genetic functions, and so on. And they even influence things like natural killer cell function, which we'll talk about shortly. So please don't go away thinking this is just a natural killer cell disease. It's not, but we use that because it's a very good model to work with. There's been a long-standing history of work in immune cells, particularly natural killer cells, Nancy being one of the forerunners in that area. And virtually all researchers have found significant immune dysfunction in those cells. And as I mentioned, we use natural killer cells because it's a good model. Ideally, we'd like to look at brain cells or heart muscle cells or whatever, but clearly that's just not feasible, and we use natural killer cells as an exemplar model. So very briefly, what actually do natural killer cells do? So as their name suggests, their role is to lyse or kill invading cells or cancer cells and so on. And they do that by releasing toxic molecules, granzymes and perforins, which then invade the attacking cell and cause their lysis or cell death. And as I said, we can measure that, and that's a very important attribute of them. And there's just results of a couple of studies that we've done. Others have replicated this kind of thing around the world, that the lysis function of natural killer cells in ME/CFS is significantly reduced compared to unfatigued healthy controls. So we thought, well, what's going on in the genetics of the natural killer cells that may tell us something about the origin of this story? And if you look at the DNA, and up in the top left in the red, the SNPs, the single nucleotide polymorphisms, we all have them, some more than others. It may be meaningful, it may not. These are known as synonymous and non-synonymous SNPs, according to whether they change function. And if they do change function, then that's a very, very important thing, because that means that there is going to be compromise in the protein that part of the DNA is going to subsequently form. Now, in natural killer cells, there's a number of, I won't go into the detail of the different types of natural killer cells. And the two that we kind of have looked at in terms of getting the data that we want are CD56 bright cells. And they're mostly associated with inflammatory regulation, but they tend to morph towards the so-called CD56 dim cell, which has got more of that lytic function that I just described about the perforins and granzymes. So when we looked at the genetics of these isolated NK cells, particularly in the TRIPS, the transient receptor potential ion channels, the left-hand column shows you a dozen or so highly significant changes that we found. If you look at the far right-hand column, they're all less than 0.05. But the second from the far right-hand side are the odds ratios, saying that these have a very, very, potentially a very profound role in the kind of pathology that we might be seeing. But of the 12 SNPs that you see on the far left, 5 of those are TRPM3. So that really stood out to us and said, well, there's a red flag. Why don't we look further into the TRPM3 to see what's going on? Now, as it turns out, the TRPM3 gene has more isoforms than any other TRP in biology. So you could draw a long bow and say, well, there's perhaps more potential for SNPs to affect a greater array of TRPM3s and hence have a greater impact on function. That's just a hypothesis. We'll look at that over time. But if you recall from last year, the other name that TRPs are given is threat receptors, because their fundamental role is to deal with incoming threats that the organism has to deal with. And if you look on the far left, this slide may be familiar. Anything that impacts in the form of a threat to the human body is dealt with by these receptors through calcium signalling. So if we go down the list, osmotic pressure changes, volume changes, stretch changes, the kind of things you see in physical exercise, for example, and we'll come back to that. Vibration, such as travel. How many patients enjoy travelling in trains, trams, buses, aeroplanes? The answer is not very many. Temperature, and we'll use that as an example later on. Injury, surgery, any of those factors have to be dealt with as a threat, and these channels manage that. Biological substances, antibiotics, allergens, infections. Doesn't really matter what the infection is, could be an enterovirus, could be a bacterium, whatever. Infections are dealt with by these receptors in terms of maintaining homeostasis. And irritants, and for example, perfume, cigarette smoke, fuel, vapours and odours and so on. But what happens is that these receptors, there are a 6-transmembrane mechanism, and the ion channel is between element 5 and 6, which allows calcium into the cell. And it's that second messenger function of calcium that impacts on every system in the body. Every tissue, every cell, every tissue in the body, in every organ system, has these TRP receptors to a greater or lesser extent. Now, two things about that. One is that calcium is in a much higher concentration outside the cell than inside the cell, many hundreds of times, if not thousands of times greater outside, and that's the calcium ion, 2+. So there's a much higher concentration outside. So the object of these receptors is on stimulation to allow the calcium ions in where they can do the work they have to do. As I said, biochemical pathways, genetic regulation, and so on. The second point, which I'd just like to make, and we can talk about this later, is that the TRPM3 receptor we also call that a store-operated calcium channel. I can explain that more later, but it is the predominant calcium store-operated channel in the glial cells in the brain. They're the support cells, the oligodendrocytes, microglia, and so on. Now, neurons, the kind of main nervous system, as it were, rely on what are called voltage-gated channels. They're excitable cells. They have to be excitable to transmit a nerve impulse. But the glial cells work a little bit differently. We call them non-excitable. That's not entirely true, but they function more on these store-operated calcium channels. And if you see an article by Papanicolaou earlier this year on store-operated calcium channels in glial cells, you'll see that the predominant store-operated channel for calcium transmission in the brain is the TRPM3 receptor, the TRPM3 ion channel, but also located in the pancreas, in the heart, and so on. And if we have time, we can talk about the physiological manifestations of what that might be. So, as I mentioned, we looked for the TRPM3 on NK cells, and yes, there they were. But, and I don't want to get too bogged down into the data, but there are differences between the so-called CD56 bright and the CD56 positive dim. But enough to say that there is a significant reduction in the numbers of TRPM3 receptors. And we can say, well, why is that so? Yes, the SNPs are probably associated with that, but there is something about the way the protein is changed that means that it does not finally express as the receptor on the cell. But bear in mind, they're not only on the surface of the cell, they're in mitochondria, the endoplasmic reticulum, and so on. So they actually manage the store, hence the name, of calcium within the cell. And that's a very fundamentally important point. So there's a number of different types of calcium channels. Again, I won't go into that in detail. But the way we look at this, because calcium can come into the cell through these channels, they can also be released from mitochondria, from the endoplasmic reticulum. It's important to block those when we do the experiments on them. And we use drugs like 2-APB, thapsigargan. I won't go into the detail of that, but they basically block other movement of calcium. So then when you do something to the cell, you know that you're getting a very kind of pure response from the intervention that you've made. Now, uniquely, the TRPM3 receptor, what we'd call the natural ligand, the substance that actually activates this receptor, is pregnenolone sulfate. So we use the abbreviation PregS. So the experiments we used involved pregnenolone sulfate. I'll skip through some of the hard data. But the question that now you're probably all asking is, if we're able to manipulate TRPM3 function using different drugs, where does that take us in terms of therapeutic intervention? And you would be absolutely correct. I'll try and just stick to the sort of take-home message here, but it was very interesting, and we got some unusual results, that we didn't specifically get a good response to PregS, right, to pregnenolone sulfate. So there was something going on, probably as a result of the change in the structure of the receptor, but it was with thapsigargan, which is one of those blocking agents, that we saw the most response. Again, differences between what we saw in bright cells and what we saw in dim cells. And the way we measure that is in terms of the calcium signal. And this is the calcium, so-called under the curve. The far right-hand peak there, the shaded area, is the calcium that's flowing into the cell that we can then measure through flow cytometry. So this is really interesting, that when we applied pregnenolone sulfate on its own, there was no change. And you think, well, that's probably fair enough because the cells aren't responding all that well, but yet they're responding in healthy controls. But when we added the thapsigargan and the pregas, we actually got a disproportionate response in the patient group. And we're still trying to work out the explanation for that. But that does tell us that there is a very complex mechanism happening within the cell involving calcium signalling that we need to know more about. And we need to know more about that because it may have a fundamental role in the pathology of this illness. Now, I mentioned before about how different organ systems and how different responses to threats occur. This is just one example, but for the sake of time, we'll stick to one. So this is temperature exposure. So different trips respond and activate to different temperatures on that thermometer, as you can see. So trip A1, down the far left hand, responds and activates to very cold temperatures, whereas trip V2, the far right-hand side, would respond and activate to very hot temperatures. Now, interestingly, patients do not respond well to certain temperature changes. With some patients, it's more at the heat end of the spectrum. With some patients, they react worse to the colder end of the spectrum. And some are in the middle. And if you look at trip M3, it kind of occupies the middle ground. So the reason I put this in is as a metaphor for what may be happening in other organ systems, in other physiologies, that all contribute in some way to the very, very complex picture that we see in ME/CFS. So this work is ongoing. As I say, the lab is continuously working. That's the team. I'd like to thank and acknowledge all of them, and particularly our donors, and to particularly acknowledge the Queensland Government, who have supported us for a long time. Thank you.
Don, thank you very much. It's now open for questions, of course, so please pin his ears back. Yeah. Yes.
Thank you. Rekha Iyer from Stanford University. I was wondering, now that some whole genome sequencing efforts are starting to be done in ME/CFS, do you have a sense of how frequent variants in this family are in other ME/CFS cohorts relative to the general population?
Yeah, look, can I just say the limiting factor for us is the cost.
Sure.
Whenever we do this, we usually don't get much change out of $100,000. So, and that's, say, around 100 patients or something like that. So we're still limited in how much we can do in that area, but you make a good point.
Right, I meant from other groups that have started to do whole genome sequencing. And
I don't know what they're doing in this area.
Or how it might be in the general population, because there's now a lot of whole genome sequence data available from healthy populations that you might be able to compare your variant frequencies and see if they're really at sort of a broader scale.
Yeah, that's a good point. We're still grappling, though, with the fundamental pathology that, yes, you may have these SNPs. Now, why is it that some people get sick and some people don't? Because
right.
If it's in your DNA, it's in your DNA. And it probably, and I'm speculating here, so please note that there's probably an accumulation of stressors, whether it's injury, whether it's infection, whether it's exercise, overtraining. There's probably a number of factors there that are involved. That's all I can say.
Sure. Thank you.
Okay, I think there's another one here. Yes, yes, sir.
Thank you for a brilliant talk and some very exciting data there. I was wondering, sorry, I'm just here. Have you seen, is there a sort of a correlation between the calcium receptor dysfunction and the severity of disease? Have you tried to sort of link the two at all?
Look, that is also a good question, and we are looking at a severe group of patients, and what we do with the severe group is to visit them in their own home. They simply can't get out of the house, so we have a protocol where we go into the home. We try and reproduce the same protocol as in the lab in terms of we take bloods and so on, but that would be the next step that we would like to do, is to see if there is a correlation with severity. But again, cost will impact on those sort of studies, but that's exactly the right question to ask.
Two more quick ones. Yes. Oh, sorry. Yes.
As a clinician, would you, I'm a clinician, would you be able to comment on the use of calcium channel blockers for patients with this illness? Relatively cheap drugs, easily obtainable?
Watch it, Don.
Obtainable?
Watch it.
Thanks, Ros. Look, that is also a really perceptive question. Now, I mentioned before that excitable cells use voltage-gated channels, right, for their calcium signalling. Non-excitable cells, such as, I don't know, immune cells, we'll take that as an example, pancreas cells maybe as well, use store-operated channels, such as the trip ion channels. Interestingly, when you use drugs like nifedipine, which are calcium blockers for the excitable cells, the voltage-gated channels, you get a paradoxical effect in trip channels. Now, we don't know why that is, but the thing about calcium blockers is they do exactly that. The problem in this disease is the calcium is not being entered into the cell. It's already being blocked by what we think are abnormal receptors. So we need a mechanism, and look, again, I'm speculating, so take that on board, that it's probably very hard now to change those receptors that have ended up being damaged, probably because of those threats overlaid on those SNP predispositions. But on a maybe, just maybe basis, we could stimulate neighbouring trips that have similar functions. For example, in the temperature chart, you saw there's some overlap. Maybe we could trick some of the, if not an M3, maybe an M2 or an M4 or something, to take over that role, using whatever the drugs are that they respond to. That's a really hairy area, and as I say, that's speculation, but that's as much as I can really say about that question.
I was happy to be able to hear your talk this morning. As you may recall, our laboratory has also focused for many, many years on natural killer cell function. We, in fact, have this disagreement that maybe during this meeting we could discuss, and that is we find that the lytic proteins, granzyme and perforin, are decreased in the natural killer cells from patients with ME/CFS. Your laboratory has reported that perforin is actually increased, and I've never been able to to understand that finding, and it would be interesting to discuss it with you.
That's going to happen. We do see paradoxical results in some areas, but I think the key point here is that calcium is essential for the manufacture and processing of those lytic granules and so on, and the release from the cell. So taking the broad picture, we still believe that calcium has a fundamental role in that mechanism. Thanks.